Chapter 6, DNA. We just finished off work on Mendel and genetics and how traits are passed on, but this is going to be more of a discussion of the physical aspects of how information is stored in cells and how that relates to DNA. And today will be a lot of history related to the discoveries surrounding DNA. First off, what is DNA? Well, it's a very long molecule with a very long name. Deoxyribonucleic acid is a special molecule found inside of cells that carries the information in a code to create proteins. These proteins then influence basically all aspects of our life. They're either components of cells, or messengers between cells, or regulators of processes that occur within cells. Again, deoxyribonucleic acid. Our first scientist in our history is going to be Friedrich Miescher, or his full name would be Johannes Friedrich Miescher-Rusch, a Swiss scientist who in 1869 isolated a substance from cell nuclei that he called nuclein. And he also, when he did chemical analysis on it, discovered that it had a lot of phosphorus in it. He hypothesized that this substance could be important in fertilization, and he was correct in that hypothesis because it is. As far as Miescher goes, background information, most of his family were physicians, and he was going to become a physician, but when he was young, he had a very bad bacterial infection that ended up causing him to lose his hearing, not his ears, and he didn't think he could be a good doctor if he couldn't hear his patients, so he went into research instead. Miescher had a student, and he had other people that worked in the lab with him. And the two of them, Miescher and his student Richard Altman, isolated some substances from the nuclein. One was a protein molecule, and the other was an acid molecule. And they thought that the acid molecule might be important to kind of continue research on, so they began more work on the acid molecule. And it was his student, Richard Altman, who came up with the name nucleic acid, and it is still the term that we use today for this acid molecule inside of cells. Albrecht Kassel, or Ludwig Karl Martin Leonard Albrecht Kassel, was a German doctor and scientist, and he identified another component of nucleic acid. He was working with the nucleic acid and breaking it down chemically, and he discovered 5 bases that are part of the nucleic acid molecule: guanine, cytosine, thymine, and adenine, and also another one called uracil. As a side note, he also discovered some amino acids, histidine, thymic acid, and agmatine, but you don't really need to know that. Additionally, you don't really need to know these dates, nor do you need to know their full names of all of these scientists. A last name will be sufficient. Phoebus, I love this name. Phoebus Aaron Theodore Levine, who looks like a very studious little scientist, was born in Lithuania, which at the time was Russian. His family grew up in St. Petersburg because they moved there, and he earned his medical degree there. However, it was sort of a turbulent time at Russia here at the turn of the century, 19th to 20th century. There was a lot of anti-Semitism occurring. His family was Jewish, so obviously that was a concern. So the family ended up leaving Russia and emigrating to the United States, where Phoebus took classes at Columbia University. And that's where he met Kassel, and so he started working on the nucleic acid molecule as well. In 1905, he accepted a position as head of the biochemical laboratory at the Rockefeller Institute in New York, and he continued working on nucleic acid. Chemically, he was able to isolate the sugar components of nucleic acid. He discovered ribose in 1909, and he discovered deoxyribose, which is the other sugar, in 1929. And 20 years between those discoveries, which may seem odd, but if you look at the chemical composition down here of these two sugars, deoxyribose It's just missing an oxygen compared to ribose. That's the only difference between the two. So not too hard to believe why it took so long to kind of figure out that those were different. He also worked on the chemical structure of DNA and discovered that it was made of a recurring subunit, a subunit that he gave a name to, which is actually on the next slide. But the subunit was consisted of a phosphate group, a sugar group, and a base, one of the 4 bases that Kossel had discovered, that adenine, thymine, guanine, and cytosine. And those 3 things, the phosphate group, the sugar group, and the base, he called a nucleotide, okay? And he hypothesized that DNA, there were, at least 4 of these units in DNA, because there were 4 bases, the adenine, the thymine, the guanine, the cytosine. And he called those group of 4 bases tetranucleotides. And this was his fatal flaw. He thought that DNA could only be 4 bases long. And because it was only 4 bases long, he didn't believe that it was complex enough to actually store genetic information. And so poor Phoebus gave up on his research on DNA, basically, and began working on some other protein that he thought might be good for genetic information. But he was wrong. Poor Phoebus. Right, Erwin Chargaff. Chargaff was an Austrian biochemist who also immigrated to the U.S. during a time of a lot of immigrations occurring. And he took a position in the biochemistry department at Columbia. And Columbia University and the Rockefeller Institute were both kind of epicenters of a lot of DNA research activity. And he discovered that when he isolated chemically DNA, the number of guanine units, like the amount of guanine, always equaled the amount of cytosine, and the amount of adenine always equaled the amount of thymine. All right, so he discovered that these two, guanine and cytosine always seem to go together, and adenine and thymine always seem to go together, which meant maybe that they were paired in the structure. All right. Rosalind Elsie Franklin was a woman scientist at a time when girls didn't really become scientists. And she had a lot of trouble kind of convincing her dad to let her go into the sciences, which she finally was able to do. And she got a PhD at Cambridge, which, is no small matter. And from there, she moved to Paris, where she worked on X-ray diffraction techniques and learning how to shoot X-rays at objects and take images from the bouncing off of the X-rays. She got good at this, and she then was offered a position at King's College in London, where she began working on DNA. And she had a hard time at King's College and in most places where she was around, because the men of the time, most of them, really didn't believe that women should be in the sciences. This particular colleague, Maurice Wilkins, treated her as an assistant, never treated her as an equal, was not very nice to her. The university itself still wasn't very forward-thinking. The dining hall was male only, so she couldn't eat at work where everybody else ate. She had to bring her own lunch in and either eat in the lab or eat in the park or, out in the garden, whatever. She couldn't eat with the rest of her colleagues, which is sort of ridiculous, but there you have it. Anyway, she, in 1951 and 1953, she came really close to solving the DNA structure issue. She took this picture using X-ray diffraction. She took this picture of DNA, and it's basically from like a top-down view of DNA. And seeing these cross images, seeing the circular structure and these cross images, led everybody pretty quickly to realize that DNA had a spiral structure, because these are the cross bits of the ladder across the spiral, so it was like a spirally ladder. And she was putting together a paper which she was going to publish, but Wilkins, that colleague that didn't like her very much, ran off with her picture and showed it to Watson and Crick, who were 2 scientists working on it at Cambridge. They immediately recognized the importance of the image, immediately put all the other information together and came up with a model. And they ended up publishing in the same journal that Rosalind Franklin was publishing her picture and information. And they actually ended up getting all the credit for the double helix structural shape of DNA and the model of DNA, and ended up winning the Nobel Prize. And poor Rosalind didn't really get very much credit. And even within her lifetime, she really didn't get very much credit because of her work with X-ray diffraction. Early X-ray research, X-rays are radioactive. I mean, there is a small amount of radioactivity associated with X-rays, and so that's why they offer lead, aprons and stuff to people that work around X-rays or that are getting X-rays done. But at that time, they were even more radioactive because it was early. They were just getting started, and they didn't know how dangerous they could be. And Rosalind Franklin actually died of cancer at a fairly young age, unfortunately. Okay, here's their model, Watson and Crick's. And it consists of the outside of the structure, like the outside of the ladder, consists of phosphate and sugar all linked together. And then the inner kind of ladder steps, the rungs of the ladder, consist of base pairs linked together. So like an A linked with a T or a G linked with a C. And those base pairs followed Chargaff's rules because Chargaff discovered that adenine always had the same amount of thymine and guanine always had the same amount of cytosine. So they always paired those two together. So adenine was always with thymine, thymine with adenine, and cytosine with guanine, equally paired together. Here's another picture. Twisted ladder. Again, this gray outer structure is where the phosphate and the sugar groups are, and the cross rungs are where the bases are located. The bases, you'll notice here, are connected to the sugar. The phosphates just link the sugars together. Knowing how the bases pair, from what we know from Chargaff, you can actually look at half of a DNA strand and know what the other half has to be. Okay, because G always pairs with C and A always pairs with T. We know that this should be A, T, G, A, G, T, C. Ta-da! Fairly simple. And this overall pairing really helps scientists figure out how DNA copies itself, because basically all that has to happen for DNA to make a copy is for it to sort of unzip down the middle and use each half as a template to add the new base pairs in according to Chargaff's rules. And then really simply you end up with 2 strands of DNA. It's very quick. So you can see how that works here in this picture. You've got stranded DNA, this is just a little chunk. It unzips. New base pairs add in according to Chargaff, A with T, G with C, all on either side, until you end up with 2 DNAs that are exactly the same, because each old half acted as a template for the new bits. Pretty cool. There's a lot more to this, and we watched some videos in class that illustrated this, and I think that posted those videos as well, so hopefully the TED Talk goes through how crazy this process is. But this is really simplified, and it's a way that I want you to know for now. DNA is a long molecule. In bacterial cells, it just usually forms a loop, just a circular structure. In prokaryotic cells, but in eukaryotic cells like ours, DNA is located inside the nucleus. It's usually, because it's so long, it has to be kind of tightly wound up around other proteins and organized just to keep track of it and to make sure that the cell can, when it's time to divide, that the cell can organize those and move them and get them so that each new cell gets the right amount, all that kind of stuff. So there's a lot that goes on in organizing DNA. Here you see DNA by itself, just the double helix. Then it wraps around these protein structures and ultimately forms these even more further coiled protein structures called chromatin. And then these wrap even more to ultimately form a chromosome. You can see it again here. DNA wrapped around proteins, coils some more. This is called chromatin again, this section when it's coiled with the proteins. And then that coils even more until eventually you get the chromosome. And remember, chromosomes consist of two sister chromatids that are exact copies of each other, and they get divided in mitosis. This is a chromosome only happens when the cell is about to divide. So DNA with its associated proteins is called chromatin. And when the cell is ready to divide, the chromatin is packed into two chromatids, which form the chromosomes. And when the cell actually divides, each chromatid gets separated and given to the two daughter cells. And that is all. The videos you can see posted online, either on the portal, and I may put some on Sophia as well. But that is all, folks.